All right, the next talk is from Eirini Schoinas, Why Cortical Implant Patients May Never See the Stars.
I'm here to talk about why cortical implant patients may never see the stars. My name is Eirini Schoinas, and I did this work at the University of Washington. And this is in collaboration with Leili Su at the Institute of Bioengineering at the University Miguel Hernandez in Spain. So blindness and visual impairment affects millions of people worldwide, and this has a profound impact on their lives. And one of the ways that researchers are addressing this is with visual implants that can be placed along or at different locations along the visual pathway. So in the retina or the LGN, or in this case, I'm talking about the cortex. So specifically, I'm going to be talking about tiny depth electrodes, which are being worked on mainly by two main groups, which is Troik in Illinois and Cordavis in Spain. And Cordavis is what I'm talking about more specifically. So this is electrode this array is a 96-channel Utah array that consists of 96 millimeter and a half electrodes, and they're very thin, and they're implanted, like you can see in that picture there, into the cortex near V1 and V2. So specifically, Leili was working with one patient, that I'm going to talk about, with the implant located about there near the border between V1 and V2. And the electrode grid looks like this, just colored by spatial location. And what she did was each electrode was individually stimulated above threshold, and this stimulus was a charge balance cathodic first biphasic stimuli at 0.4 milliseconds per pulse at 300 hertz over 166 milliseconds. And what the patient did was use their finger as a fixation reference for the center of the visual field, and then they drew this phosphene, which is what they saw in their visual field, on a tablet, and then also described this. And from this, we get where these phosphenes are showing up in the visual field, and then also what they look like, which would be, this is just an example. And what she found was that the phosphenes were replicable, especially given, just keep in mind generally, these are all things that are drawn by a completely blind person. So anyway, here's an example of 4 drawings from the same electrode being stimulated, and then with a verbal description, you're able to turn that into something clearer. And she also did a repeated identification experiment where 2 to 4 electrodes were picked out and then stimulated at random, and then the patient had to pick out which electrode was being stimulated, and there was near ceiling recognition on that. So not only are these phosphenes replicable, but they're also, many of them at least, are distinct from one another. And ultimately, what we see from this is that these phosphenes ended up varying a lot. So they're not just simple little like blobs or stars, but this is just a subset of the ones that we had, but already you're seeing they're really kind of all over the place. And some of these, like the 2 lines on the bottom, those 2 electrodes are right next to each other. But then there's a similar motif, but they're already at different orientations. Or like the orange kind of like star-looking one, and then the one above it, those are also next to each other. But either way, as you go to all these different electrodes, you're getting a lot of different things. And some that look similar that are far away from each other, some that look similar that are close by. You can't tell what they're going to look like just by their spatial location. And really, this kind of presents a problem because most models are assuming that tiny little electrodes are going to produce small dots or stars, but clearly, at least for this patient, this is not the reality here. And this leaves us with some questions, mainly why are these complex phosphenes occur, which I'll talk more about in a second, and also how can we model them? So can a simple model of the cortex explain this phosphene variation? So this model is defined as follows, basically. First, you have to map from the cortical location to the visual field locations. And so basically, you can see the cortical magnification here, which basically just means that if a large portion of the cortex maps to a very small portion in the center of the visual field. So basically, if you stimulate a location here in the cortex, you're going to see a phosphene there in the visual field. Next, for each pixel, we want to define what orientation and or they respond best to. So each pixel is basically just, in this case, a clump of neurons that responds in the same way. And we want to define what orientation they best respond to, and then also the ocular dominance columns, meaning whether they respond more strongly to input from the left or the right eye or somewhere in between. We then use the general Ringosh model to define simple, complex, on-center and off-centered cells, and you can just change the parameters in this and then match it to the neurophysiological data to match what happens in the cortex. And we bias it towards on because when you're stimulating, you're generally going to see a bright phosphene. And then here's some examples of the receptive fields that you get when you're changing the eccentricity in the ocular dominance, and you can see that as the eccentricity, for example, increases, the receptive fields get bigger. And then the final phosphene is created by a linear sum of the receptive field profiles weighted by the current spread. And you can see as you're increasing the electrode radius, the phosphenes are getting bigger, but also importantly, small electrodes are producing irregular phosphenes. And to understand more why this is happening, I'm going to talk more about the orientation map. So generally, if you're placing an electrode on a patch of neurons that all responds to the same orientation, you're going to get a phosphene that kind of mirrors that. And you can see that with the green one, it's kind of all in the same orientation. On the other hand, if you place an electrode between or on electrodes or not on neurons that respond to multiple orientations, you're already going to get something kind of more complex, like this yellow one. Furthermore, we know that axons in the cortex exist that carry signal between patches of neurons that respond to the same orientation, so between the two green patches here. And in this case, we're placing the green electrode down. That's an actual electrode. So what happens when you place one there over an axon? Well, what can happen is you can get a response as if you were also stimulating the blue location, but you're not. The electrode is only in the green spot. And you can get something like the bottom phosphene, which is even more complex. So keeping this in mind, we did 4 main steps to attempt to now model the actual reported patient phosphenes. So first, we had to process these patient images, meaning like we have an original drawing. This is already slightly cleaned up, but furthermore, we wanted to thicken the lines and so on, so when we're doing correlations, we can actually match the structure and also fully match their verbal description, like filling in the phosphenes in, and so on. And then if we had a more complex phosphene, we also split it into different subcomponents so that we could attempt to individually fit for the different subcomponents as well. We then wanted to simulate the individual phosphenes. So we simulated 1,000 different phosphenes at various electrode radiuses and eccentricities, and we found the shift, scale, and rotation that produced the strongest correlation with the basis images or original drawing. And then we saved the top 24 phosphenes, making sure that they weren't too similar with each other so that we would have some variation there. And if we had multiple components, we just split that 24 among the different components and with the original drawing as well. We then also wanted to combine the subcomponents together. So we had 24, we did 24, choose 1, 2, and 3, and we found all the different combinations. And when combining them, we used linear regression to find the weights for each subcomponents by minimizing the error with the original drawing. And we saved all of these as well. And like for this one, you can see like with one phosphene, we're not really capturing the structure of it very well. But as you add 2 or 3, you're improving on that a lot. So finally, we also want to determine how many sites we actually need because that depends on the phosphene or the drawing / phosphene itself. So our goal here is to just successively add the components to predict a given phosphene until adding the next component is no longer significantly improving our fit. And what we found was at the moment, we looked at 64 patient drawings total, and currently around 37 of the drawings could be represented with just one site, 11 with 2, and 16 with 3. So here are some examples of drawings that could be fitted with just one site. So that's like simpler shapes like a line or a dot or, this slightly more complex one, which where adding 2 or 3 wasn't really helping either. On the other hand, here are some 2-site examples. So here is just attempting to fit these with just one site. And you can see you kind of are seeing the shape, but if you add a second one, you get a lot better fit, clearly. And then here are some examples of where 3 sites was best. So here's if you just try to fit it with 1, with 2, or with 3. And keep in mind, again, these are all drawings done by a completely blind person. So obviously, we're not trying to get an exact fit. We're just trying to get a general, like a structural fit where there's a similar shape and where you could see why somebody would say it looks like that. So ultimately, what we're finding here is that small depth electrodes produce surprisingly complex phosphenes. Like these shapes here, they're not just stars, they're not just little dots. And a simple cortical model, like the one I've described, can predict the variation seen across the electrodes. However, without knowledge of specific patient orientation maps, we're not going to be able to predict individual phosphenes. And this is going to, I think, make creating meaningful images with these small depth electrodes, which is, of course, the goal, especially challenging. So thank you. I want to thank my co-authors, specifically Layli Su and Eduardo Fernandez, for providing the data to us. And yeah, thank you.
Time for questions.
I'm curious about their drawings and whether they could distinguish between brighter than a background and darker than a background. All your drawings seem to be white on a black background. Are these, are they blind from birth? Do they have a concept of bright and dark or white and black?
This person, I think, is like 60, and they've been blind for around 16 years, 16 or 20 years, something like that. So, like, yeah, and but there's no light perception at this point. And I think just generally this person specifically was reporting that they were seeing bright phosphenes. There were maybe some darkness, but like the ones we're looking at, like those were reported to be bright like that. I don't know about, I don't know if there's variation between that. I think it's just bright generally, but like not, yeah, like bright, not dark.
Okay, some of the drawings, it was like the very bright white, and some of them was more like gray.
I think that would be an example of like the variation in brightness.
Thanks.
So I wanted to come to your last point about creating the perception of pattern. And I imagine your model would predict if you stimulated 2 electrodes, you would see, roughly speaking, the 2 phosphenes together. Do you know if is that a correct understanding of the way your model works? And then is it known whether that's the case when you actually stimulate this patient with more than 1 electrode?
Like, yeah, sorry. Yeah, basically, if you stimulate multiple electrodes, they just end up, or the phosphenes end up kind of on top of each other. And it's also been shown that like you have to get your electrodes pretty far apart for them to be like completely separate. And wait, sorry, what was your other question? Oh, is that the case for what's happening? Yeah, I think if you stimulate more electrodes, you're gonna get different shapes at least, like more things going on. And then if they're far enough apart, they'll be separate.
And I guess my question also included, are the patterns that they see predictable from the reports of the individual phosphenes for each alone?
I'm not sure we have data on that, but I would assume yes, but I don't think it's just a direct, like, those 2 exactly just show up. There's something else going on, but I don't have data on that, right, at least at the moment.
Okay, well, if there are no more questions, let's thank the speaker again.